So in the first video of this chapter 15, we were introducing the concepts behind C4 and CAM photosynthesis. These plants, as we talked about, are restricted, C4 plants are restricted to semi-arid habitats where there's summer rainfall, and these are the grasses, corn, and sugarcane. And CAM plants are more arid types of plants, as well as so xerophytes as well as epiphytes, plants that grow on other plants. So we spent a little time looking at C4 photosynthesis in the last video, looking at the difference in the anatomy, which is going to be advantageous here to its the physiological steps here. So we talked about how C4 photosynthesis actually involves two steps. The first step, where carbon dioxide is captured by a 3-carbon molecule into a 4-carbon molecule oxaloacetate, which is converted to malate. And then this is the transferable form from a mesophyll cell into a bundle sheath cell. So now this 4-carbon molecule enters the bundle sheath cell. Decarboxylation occurs here, where that CO2 molecule is now removed from malate. Malate becomes pyruvate, and that CO2 molecule can undergo a second carboxylation step. The first one happening previously in the mesophyll cell up here, that was catalyzed by phosphoenol pyruvate carboxylase. The second carboxylation step is the one we're already familiar with, where Rubisco catalyzes fixation of the CO2 molecule that's released here. That CO2 molecule becomes fixed with RUBP. And then we have our regular Calvin cycle happening here, resulting in 2,3-phosphoglycerates, and that's going to, of course, then move on to form two G3Ps, and so forth. We were talking about phosphoenol pyruvate carboxylase, which, unlike Rubisco, does not have the capacity to oxygenate RUBP. So it's not an oxygenase enzyme. So that makes it very efficient or very useful as a first enzyme for carboxylation of or for fixation of CO2. Let's see, so we'll be comparing phosphoenol pyruvate carboxylase and Rubisco here shortly, but not only is it not a oxygenase molecule or enzyme rather, but it also has a higher temperature optimum in comparison to Rubisco, and we'll be looking at those numbers, but somewhere on the order of 30 to 35 degrees Celsius, whereas Rubisco has a lower temperature optimum, somewhere around 20 to 25 degrees Celsius for its temperature optimum. Now we want to make sure we connect kind of what's happening in these two steps between the mesophyll cell and the bundle sheath cell with the overall anatomy that we talked about here. So in the mesophyll cells out here, these would be mesophyll cells, would be where the first step of C4 photosynthesis occurs, where CO2 is first carboxylated by phosphoenol pyruvate and converted to oxaloacetate and then to malate. Then that malate is transferred into now the bundle sheath cell, which is these cells here, and that's where malate becomes decarboxylated. CO2 is then fixed again, but this time by RUBP. So reflecting back on the anatomy here, remember we talked about the tight arrangement between bundle sheath cells and mesophyll cells. So if CO2 escapes from bundle sheath cells out into this direction, then they're then they're just going to be recaptured, recarboxylated by PEP carboxylase to phosphoenol pyruvate again. So it's a very efficient arrangement of these cells to make sure CO2 is captured, is that it's fixed to become a 4-carbon molecule and delivered to the Rubisco enzyme, where it now is highly concentrated in the vicinity of Rubisco, and we have in comparison with the concentration of oxygen. So this increases the, it's supposed to be an increasing arrow there, increases the CO2 to oxygen ratio at the site of Rubisco. Rubisco, let's spell that correct. All right, now if we take a look at overall photosynthesis rates, we're looking at photosynthesis curves here, light response curves, which we've looked at before. Photosynthesis light response curves for C3 versus C4 plants. So we're looking at a radiance level. Remember, this is light in micromoles per meter square per second, micromoles of photons, versus a rate of photosynthesis, and this perhaps could be CO2 uptake, micromoles of CO2 per meter square per second. Okay, so the question is, which of these curves is the C3 plant, which of these curves is the C4 plant? Well, C4 plants have a much higher photosynthesis rate. The photosynthesis rate of a C4 plant is about 2 to 3 times greater than 2 to 3 times greater than a C3 photosynthesis rate overall. We can see, so this represents the C3 plant here. We can see that the if we look at the slope of these two lines here, remember this is where we determine photosynthetic efficiency, and photosynthetic efficiency is greater in the curve A, where C4 plant is, than in C3. Because remember that these plants are adapted to high light, so they have a large amount of machinery for the light reactions to take advantage of that high light. They also are adapted to high light intensities, so they are equipped basically to deal with the excess energy from light that may come in as a stressor. Let's see. They also have, remember this part of the curve we refer to as photosynthetic capacity, which reflects back here to the y-axis as the maximum photosynthesis rate. And so you can see that the photosynthetic capacity of the C4 plant is also much higher than in the C3 plant. Again, because they are making more ATP, more NADPH, they have more of the machinery associated with the Calvin cycle. And remember also that they're concentrating higher CO2 at the site of Rubisco. So Rubisco is not CO2 limited. And so perhaps more Rubisco is synthesized and activated so that it can fix that additional CO2 that is not normally present around Rubisco in at the site of Rubisco in a C3 plant. Now, this higher photosynthesis rate in C4 plants comes at an energy cost. So when we look at C3 photosynthesis, which is a step that the C4 plant has, C3 plants use, remember, 3 ATP molecules per CO2 molecule fixed. You can see there. And then plus 2 NADPHs. A C4 plant uses the same amount, 3 ATPs per CO2 fixed, in the bundle sheath cells, But remember, that's the second step. The first step is when CO2 is carboxylated by phosphoenol pyruvate. That part of the process requires anywhere from 1 to 2 ATPs per CO2 fixed. We talked about mainly one C4 type of C4 photosynthetic pathway, but there's actually 3 different ones that are known, and some of them are, require 1 ATP per CO2 fixed, and the others perhaps 2. But this is, remember, occurring in the mesophyll. Okay, so that is a total for the C4 plant of anywhere from 4 to 5 ATPs per CO2 molecule fixed. So it is more energetically expensive. However, when we consider, these are the photosynthetic light response curves in high light, you can see that the photosynthesis of the C4 plant is thriving. But if we take this back down to low light, then the C4 photosynthesis, the maximum photosynthesis rate is going to decline. So C4 plants are most at an advantage in high light, which is during a time when the C3 plant would not even be able to photosynthesize, because what happens under high light for C3 plants, they actually have this leveling off in their photosynthetic capacity, which, remember, is photoinhibition. So despite the extra cost in energy, these C4 plants are photosynthesizing during at a light level that would be prohibitive for photosynthetic capacity to be maximized in a C3 plant. Now, if we reflect back on the usefulness here of C4 photosynthesis, remember we initiated this discussion in talking about photorespiration, which in C3 plants becomes more prevalent when the ratio between CO2 and oxygen declines. In other words, there's more oxygen outcompeting carbon dioxide for binding sites with Rubisco. And so if we look back at our two-step process here, we can see that what's happening in the mesophyll cell is is capturing CO2 so that it can be delivered to the bundle sheath cells where Rubisco is located and can have that high CO2 concentration. So what we're seeing here is, and we'll write this out, is that, we'll kind of write it in the corner here, is that in C4 photosynthesis, CO2 fixation is in basically 2 steps that are separated in space between 2 cells, in other words, between the mesophyll cell and the bundle sheath cell. And what we're going to see next, in the next video clip, is that in CAM photosynthesis, those 2 steps are separated in time, day versus night, instead of between 2 cells. So that's where we're going to pick up from this point in the next recorded lecture.